Hello and welcome, my dear students. Today we are going to discuss the cell biology chapter in terms of the NEET revision, and we are going to highlight the most important points from NCRT that you must give more emphasis on. So, mainly, our target is to discuss those points which are very important in this cell biology. So, let's start from the first topic, that is the discovery of the cell. So, from the discovery of the cell, you must remember, if you are asked in general, who discovered the cell? If you are asked in general, who discovered the cell, so the answer will be Robert Hooke. Robert Hooke. The answer will be Robert Hooke. But you must also remember that Robert Hooke discovered the cell, discovered a dead cell, dead cork cell. And when was it? It was 1665. So he discovered a dead cork cell, and the name cell he gave became misnomer, because actually cell is not dead. Cell contains a lot of metabolic machinery inside, but Robert Hooke observed the empty cork cells, so he gave the name or term cell. Now, if you are asked precisely first living cell, so the answer will be Anton von Leeuwenhoek. Anton von Leeuwenhoek. Anton von Leeuwenhoek. He described the first living cell. Okay. Then next question from this discovery of the cell will be about the cell theory. Who put forward the cell theory? And if you are asked about cell theory, then you must remember the name Sheldon and Schwann. And sometimes you can be asked about these two scientists. So remember Sheldon. Who was Sheldon? Sheldon was a German botanist who studied plant cells. He was a German botanist who studied the plant cells in 1838. Studied plant cells in 1838. Okay. And you observe that plants are composed of different kinds of cells which form the tissues of the plant. And who was Schwann? He was a zoologist. He was a British zoologist. He was a British zoologist. And this person, Schwann, this scientist Schwann, studied the animal cells in 1839. In 1839, he studied the animal cells. Okay. And he also reported that there's a thin layer on the outer side of the animal cell, which he termed as plasma membrane. So it can be sometimes asked. He observed a thin layer called plasma membrane outside the cell. So sometimes you are asked who discovered plasma membrane, then the answer will be Schwann. Okay. So these are the main points. Now, about the postulates of cell theory. Now you must know that the cell theory had one drawback. It did not explain the lineage, cell lineage, or the production of new cells. So cell lineage. Who explained the cell lineage? So cell lineage. It was explained by Rudolf Virchow. Explained by Rudolf Virchow. And what was the statement of Rudolf Virchow? It was in 1855. What was his statement? He said, omnis cellulae cellula. Omnis cellulae actually. Cellulae. Omnis cellulae cellula. And what did it mean? It means that new cells arise from pre-existing cells. New cells arise from pre-existing cells. New cells arise from pre-existing cells. Okay, these are things that you can be asked from discovery of the cell. So let's go to another slide, which is about the variation of cells. So cells usually show variation in two things. There are two types of variations that cells show. So there is variation in size and there is variation in shape. And what are the things that you are going to remember in the size variation? So in the size variation, you must first of all remember that which is the smallest cell and which is the largest cell. Smallest cell is mycoplasma. Smallest cell is mycoplasma. Okay, smallest cell is mycoplasma, and they're about 0.3 microns in their length. Now you must remember the largest cell. Largest cell is an ostrich egg. Ostrich egg. You must know the size of typical bacterial cell. Typical bacterial cell, which according to your NCERT is 3 to 5 microns. You must know about the size of RBC. So I am only highlighting the NCERT points which are very important, and NCERT numericals. So 7 microns, it is the size of RBC. Okay, so these are some important things that you must remember. You must remember. Okay. Now I can add the size of viruses, as viruses are not true cells, but you can remember their size 0.02 to 0.2 microns. This is the size of the viral cells. Most important is the size of smallest cell mycoplasma, largest cell ostrich egg, typical bacterial cell size, and size of RBCs. This is an additional point. Now among the shapes, what can you ask? What can you be asked? So you can be asked about the shape of RBCs. You can be asked about the shape of WBCs, which are mentioned in your book. You can be asked about the shape of neurons, the exact points which are mentioned in your text. You can be asked about the shape of tracheid, and diagrams are also given in your book, and the shape of mesophyll cells, and the shape of columnar epithelial cells. Columnar epithelial cells. So I am going to put forward the exact keywords that your book has mentioned. So RBC, it is round and concave. WBC, it is amoeboid. Neurons, branched and long. The key term is branched. Tracheid, the key term is elongated. Mesophyll cells, round and oval. And about the columnar epithelial cells, long and narrow. Long and narrow. So that was about the variation of cells. So we discussed discovery of the cells and variation of cell. Let's go to the next part, types of cells. This is very easy. We have mainly two types of cells. We have eukaryotic cells and we have prokaryotic cells. And we are going to list the differences among them. We're going to list the differences among them. So first of all, size of eukaryotic cell is more, size of prokaryotic cell is less. Second important thing, eukaryotic cell has nucleus, but the nucleus is absent. So DNA is packed by stones in eukaryotic cell, but the DNA is naked, which means that it's not packed by stones. Next point, importantly, there are several double-stranded linear molecules of DNA. Several double-stranded linear molecules of DNA, which we call as chromosomes. But there is single double-stranded circular DNA molecule, which is a very important point. These all points were related to the nucleus. We're going to go ahead. So here membrane bound organelles are found. You find the membrane bound organelles. So membrane bound organelles are found, but again in these they are absent. Your cyclosis or cytoplasmic streaming is present, but again it is absent. If we look at the ribosomes, they are 80s in case of eukaryotes, but they are 70s in case of prokaryotes. Okay, and what else? We can talk about cyclosis, ribosomes, and membrane-bound organelles. These are some main important points. There are some other points about the cell wall, which is mainly present in the which is cellulosic in case of the eukaryotic cells, but prokaryotes they have prepared cell wall made of peptidoglycan. But those points are not usually asked, so I will close this table right here. These are some important points. You should always look at these points. Okay. So this was about the types of cells, which was very easy. You know all about this. Now let's go to next slide, which is about the cell organelles. So how many cell organelles are there? So we actually divide the cell like this. We put the cell into non-living part, divide into two parts, non-living part cell wall and the living part protoplast. Cell wall may be absent in certain cells. Then the protoplast we divide it into cell membrane and protoplasm. Then protoplasm we divide into cytoplasm and nucleoplasm, which is inside nucleus, outside nucleus. Cytoplasm into organelles and cytosol, and organelles into amembranous, without membrane, single membranes, and double membranes. Now there can be a question in this classification: amembranous, single membranes, and double membranes. So amembranous organelle, what are these? This is ribosome, this is centriole, and also nucleolus. Single membranes organelles, what are these? This is endoplasmic reticulum, Golgi apparatus, lysosome, and vacuole, collectively called endomembrane system. These are collectively called endomembrane system because their activities are closely linked. And double membranes organelles, this includes mitochondria and plastids, chloroplast more precisely. So this is basic division of the cell. So we can have cell wall outside and all living matter protoplast. Protoplast has the outer cell membrane and the inner protoplasm. Protoplasm is divided by nucleus into cytoplasm and nucleoplasm, and cytoplasm is organelles and cytosol, organelles into amembranous, single membranous, and double membranous, which are all listed here. Next, we're going to discuss the plasma membrane. So important plasma membrane, we are going to only discuss about the important points about the plasma membrane, which can be asked in our examination. Okay, so plasma membrane. So what is the first important point that it can be asked? Okay, so it is also called as plasma lemma. It's also called as plasma lemma. Okay, this is the first point that you can be asked. Now important, this the term was given by Singer and Nicholson, Nagili and Kramer, sorry. Nagili and Kramer gave the term. And its structure was explained by fluid mosaic model. Fluid mosaic model, which was given by Singer and Nicholson. Singer and Nicholson. Very important. In 1972. This is also important. Okay. Now, what is the first question that you can be asked? So first question is the composition. What is it made up of? Composition. So composition, number one, it contains lipids. Number two, it contains proteins. Number three, it contains carbohydrates and also water. So this is the complete composition. Okay. Now about lipids, you can be asked what are the type of lipids found. So there is a bilayer of lipid. There are two layers of lipids, and which lipids? There is a bilayer of phospholipids. Phospholipids, and a phospholipid molecule is like this. It is having these two ends. This is the bilayer. This is the head. This is the hydrophilic head of phospholipid molecule. Hydrophilic head of phospholipid molecule, and these are hydrophobic tails. These are hydrophobic tails, and you must note that tails are embedded inside while head is projecting outside. Now about the proteins. What are the types of proteins? Proteins, first of all, they are embedded like icebergs in the lipid bilayer. So lipid bilayer is the main thing, and proteins are embedded like icebergs in it. Okay. Now note carefully. They can be of two types. They can be extrinsic or peripheral proteins, and intrinsic or integral proteins. And some of them can be transmembrane also. Transmembrane or tunnel proteins. Transmembrane or tunnel proteins. Now what is the difference? Extrinsic, peripheral, they are not embedded inside. They can be easily removed. They can be easily removed. They cannot be, they are very well embedded inside, cannot be easily removed, require a lot of energy, and they span the entire thickness. They are embedded in an entire thickness of the cell membrane. For example, aquaporins, which carry the water across the membrane. So they cross the entire length of the membrane. Okay. Now important thing, this thing, carbohydrates. Carbohydrates are only present on the outer side, only on the outer face, and they form glycocalyx, a layer called glycocalyx, and they are attached to this lipid molecule or protein molecule, so forming glycolipids or glycoproteins respectively. Glycoproteins. So carbohydrates, they are attached either to protein molecule or to lipid molecule. If attached to protein molecule, they are called glycoproteins. If attached to lipid molecule, they are called glycolipids. Now important thing, some extra question. Stability of the membrane. What gives stability to the membrane? Stability of membrane is given by, is determined by. This is the important question you can be asked. It is given by cholesterol. So cholesterol is responsible for stability of the molecule. So cholesterol are rigid molecules which are present in this lipids, between the lipids and proteins, and they stabilize the membrane. And important, what is the state of membrane? Membrane is quasi-fluidic. Quasi-fluidic, half fluidic. And this quasi-fluidic nature is responsible for this cell growth. It is responsible for cell division. It is responsible for cell secretion, etc. And next important point, what is the permeability of the cell membrane? It is selectively permeable. It is selectively permeable. Very important. So this is all this about the important points which are concerned with respect to a cell membrane. Let's go a bit ahead. Let's discuss this endoplasmic. So endoplasmic reticulum, what can you be asked about? The first point that you can be asked about endoplasmic reticulum is, what is it actually? Which type of organelle is it? It is obviously a single membranous organelle. And what is it? What does it consist of? It consists of a network or reticulum of tiny tubular structures in the cytoplasm as per your NCRT. So NCRT, how does NCRT define this? Okay. So NCRT define this as network or reticulum. So reticulum is another term for network. That's why it's called endoplasmic reticulum of tiny tubular structures. We'll stick to this network or reticulum of tiny tubular structures. We'll stick to this definition. Now, one important thing. I may add here these tubular structures are of three types. There may be cisternae, which are flattened structures like this, flattened flattened structures, sac-like structures. There may be tubules which are tube-like, like this, or there may be vesicles like this. But most commonly we see cisternae. Now, important thing that you must remember, it divides the cytoplasmic space into two compartments. It divides the cytoplasm into two compartments. The space inside these tubules is called intraluminal compartment, and outside these tubules is called extraluminal. So two compartments. One is intraluminal, which is simply inside the endoplasmic reticulum, and second is extraluminal, which is simply outside. Okay. Now there are two types of endoplasmic reticulum. One is so-called smooth endoplasmic reticulum, and one is so-called a rough endoplasmic reticulum. And we'll revise the simple points. What do you mean by smooth and what do you mean by rough? Smooth appearance is smooth. It is called smooth because the appearance is smooth and it doesn't have ribosomes. So it has no ribosome on its surfaces. So surface has no ribosomes, hence the appearance is smooth. But rough endoplasmic reticulum, it has ribosomes on it. That's why we call it rough, which gives the rough appearance. Okay. Now, to be precise, we'll mention the one point that you will be asked in NEET. That is, what is smooth? What is the function of smooth endoplasmic reticulum? The function of smooth endoplasmic reticulum is synthesis of lipids and also glycogen, and also it helps us in detoxification, which is by a special enzyme called cytochrome P450. Now, what is the function of RER? The function is synthesis of proteins. It helps us in synthesis of proteins. Okay. So that was all that you must remember about the endoplasmic reticulum. Rest of the things that you are not asked about the endoplasmic reticulum. Okay. Now we'll go to the Golgi apparatus. So Golgi apparatus, first and the foremost thing, who discovered the Golgi apparatus? It can be asked. Camillo Golgi in 1898. So it was discovered by Camillo Golgi. In 1898, and he observed them as densely stained reticular structures near nucleus. So how did he observe them? Very densely stained reticular, which means network-like structures. And where did he observe them? He observed them near the nucleus. Okay. And a technique is used to study Golgi apparatus. So you must remember that technique is called silver impregnation technique. It's not written in NCERT, but it's a common point that may be asked. Okay. Now, if we talk about the structure of Golgi apparatus, it consists of these flattened and concave convex structures called cisternae. So Golgi apparatus is like this. So it consists of stack of structures called cisternae. Flattened structures called cisternae. And where are these cisternae? These are the cisternae. Okay, this is a stack of cisternae. Now remember, each stack contains 4 to 8 cisternae. One stack of Golgi apparatus that has 4 to 8 cisternae, and the diameter of cisternae is, which can be asked, is the diameter of cisternae is 0.5 to 1 micrometer. Okay, now important thing, this Golgi apparatus has two faces. It has this convex face and it has this concave face. So there is a convex face and there is a concave face. There's a convex face and there's a concave face. This is convex, this is concave. Convex face is called as cis face, or cis face or forming face, and concave face is called as trans face or maturing phase. There is difference in these two phases. So what happens is Golgi apparatus receives the raw materials from endoplasmic reticulum via this convex phase, and then the matured materials are released through the trans or concave or maturing phase. Okay, so what is the function of cis or forming phase? It receives raw materials. It receives raw materials from ER. From ER. And what is the function of trans or maturing phase? It delivers processed materials to their target sites. Okay, this is the important thing that you must always remember. Okay, now what is the function of Golgi apparatus? So first important function is packaging and processing. That's why it is sometimes called as baker's body. So the first important function is processing and packaging of raw materials from ER. Processing and packaging of raw materials which it received from ER, endoplasmic reticulum. Number 2 is synthesis. This one is asked many times. Synthesis of glycoproteins and glycolipids. Synthesis of glycoproteins and glycolipids. This one is very important. Okay. And number 3 is formation of cell plate during the cell division, and formation of acrosome in the sperms, formation of root hairs, etc. So there are various structures which are formed with the help of Golgi apparatus. So formation of cell plate during cell division, formation of acrosome in the sperms, formation of root hairs, all these extra functions, they are also performed by Golgi apparatus. But mainly you are asked about these two functions which you should always remember. Okay, so let's go to lysosomes. So what are lysosomes? Lysosomes, they are called as suicidal bags. So actually they are vesicular structures which are actually formed by Golgi apparatus. Okay, so these are vesicular structures. It's a vesicular, round vesicular structure formed by Golgi apparatus. Formed by Golgi apparatus. Formed by Golgi apparatus. So Golgi apparatus is organelle responsible for formation of ribosomes. Now, what do these vesicular structures contain? They contain a mixture of hydrolytic enzymes. Hydrolytic enzymes. And what are those hydrolytic enzymes called as? They are called as acid hydrolases. You must always remember the pH of lysosome is round about 5. So it contains acidic pH and the enzymes are active at that pH. Okay, that's why they are called acid hydrolases. Now there are various examples of these acid hydrolases. There are carbohydrates, or what we say amylases. There are lipases. There are proteases. There are phosphatases, etc. So there are different types of acid hydrolysis which are found in these lysosomes. Okay. Now, important thing about these lysosomes. What are they useful for? They are useful for intracellular digestion. So what they help in? They help in intracellular digestion. Normally digestion occurs extracellular in human beings in the digestive tract, but if the digestion has to happen inside the cell, the organelle involved is lysosome. Intracellular digestion. They also help in autophagy. Autophagy. Autophagy is the removal of, digestion of worn out cell organelles. So there are some cell organelles which get damaged. They are then digested or removed with the help of lysosomes. That's the important function of lysosomes. As per NEET, these are the only points that you may be asked. Okay. And they also show there are different types of lysosomes. You must really remember there are different stages of lysosomes. You must, I will just say the names, you can read them if you want to. There is a primary lysosome, there is a secondary lysosome or heterophagosome, there is a tertiary lysosome or what you call as a residual body. There's a tertiary, and there is autophagosome. These are the 4 types of lysosomes. You can read about these types. Okay. Now we'll go to next organelle, that is the vacuole. Okay, now what is vacuole? Vacuole is again a membrane bound space found in cytoplasm which help us in storage of various materials, especially in the plants. So it's a membrane bound space, membrane bound space in cytoplasm, typically defined as membrane bound space in cytoplasm. Now, to be precise, there are 3 main types of vacuoles. 3 types of vacuoles. 3 types of vacuoles. One is so-called sap vacuole. Second is so-called contractile vacuole. And third is so-called food vacuole. Remember, the most important among them is the sap vacuole. Sap vacuole is found in plants and animals. It is found in plants and animals. Plants and animals. Okay. Now, the type of now animals, they have small numerous vacuoles. Small numerous sap vacuoles. But in plants there is difference. In young plant cells, in young cells, again we have these small numerous vacuoles. But in mature cells, there is single large vacuole. Single large vacuole which occupies 90% volume of the cell. Which occupies 90% volume of the cell. And this single large vacuole is called as central vacuole. This single large vacuole is called as central vacuole. Okay, you must remember this. Now, what does this vacuole? An important thing, its membrane is called as tonoplast. Okay, I will note that here. Membrane of vacuole is called tonoplast. Membrane is called as tonoplast. This is a question which is asked many times. This question is asked a lot of times. Now, what is the function of vacuole? What is the function of vacuole? If this sap vacuole, it is storage of the materials against concentration gradient. Okay, so storage of materials against concentration gradient. Storage against concentration gradient. Storage against concentration gradient. Now, if someone asks us how is this possible that they are able to store materials against concentration gradient, that is with the help of the transporters present in the tonoplast. Okay. Now, about this contractile vacuole. Now remember, this contractile vacuole is found in these amoeba. It is found in amoeba and other related protists. Amoeba and other related protists, which are usually freshwater. And what this contractile vacuole, obviously it is able to contract. And what is the function of that contraction? The function of that contraction is to remove the water from the cell and to maintain a proper water level in the cell, what we call as osmoregulation. So what does it help in? It helps in maintenance of water level, that is called as osmoregulation. And also, as it is pumping out the water, excess water from the cell, in that water excretory wastes are also removed, so it also helps us in excretion. So it helps us in two things: osmoregulation and excretion. So there are two functions of the contractile vacuole: osmoregulation, that's maintenance of water level, and excretion, removal of the wastes. Then there is this food vacuole. Food vacuoles are typically the vacuoles formed after phagocytosis. So they are the vesicles formed after phagocytosis. After phagocytosis, every one of you must know phagocytosis. Phagocytosis, cell eating. For example, this is a cell and this is a food particle, green color food particle outside. Now cell eats this by this process called as phagocytosis, like this. It is this by the process called as phagocytosis, and this inner particle now formed is called as food vacuole. Okay, these are all questions that you can be asked about the vacuoles. Now let's go to mitochondria. Let's quickly revise mitochondria. So mitochondria, everyone will be familiar of mitochondria. Mitochondria is also called as powerhouse of the cell, or some people like to call it as bioblasts. Okay, so they were discovered by Kolliker and the term was given by Benda. It is not asked, still you can remember they were discovered by Kolliker, and जो term had the term mitochondria, it was given by Benda. Okay, Benda. Now, you can be asked about the number of mitochondria, which you should always remember depends on activity of the cell. So, number of mitochondria depends on activity of cell. Number of mitochondria is directly proportional to activity of the cell. If the cell is more active, then it will have more mitochondria. You can be asked about the shape of mitochondria, which is sausage or cylindrical. Cylindrical or sausage. So sausage is better term. Now you can be asked about size of mitochondria, and in the size you can remember the length of mitochondria is about 1 to 4.1 micron, and the breadth is about diameter is about 0.2 to 1 micron. Okay, so this sausage shaped mitochondria, you can measure its length and you can measure its breadth. Okay, now there are two membranes in each mitochondria. If we look at this mitochondria, there are two membranes. There's an outer membrane and there's an inner membrane. And important thing is inner membrane is folded, and these folds are called as cristae. The folds are called as cristae. So if we talk about the structure of mitochondria, so structure is quite simple. It has two membranes. It has two membranes. Outer smooth membrane and inner folded membrane. Outer smooth membrane and inner folded membrane. And you can be asked what are the folds called as? Folds are called as cristae. They are called as cristae. Okay, now remember, if we take this as one crista, if this is a crista, each crista has these racket shaped particles. Each crista has these racket shaped particles which are responsible for ATP synthesis. And what are they called as? They are called as F0 F1 particle. So each crista contains these F0 F1 particles. or what we call as oxysomes, or elementary particles, or Fernandez-Moran particles, FM particles, or Fernandez-Moran particles, and they are responsible for ATP synthesis by process called as oxidative phosphorylation. So they help in ATP synthesis by process called as oxidative phosphorylation, called as oxidative phosphorylation. And you must always remember this inner membrane is less permeable than outer membrane. This is also important. Now this inner space in the mitochondria is called as matrix. It's called as matrix. And you must always remember matrix has certain important things. It contains enzymes of Krebs cycle except succinate dehydrogenase. It contains 70S ribosomes. It contains double-stranded circular DNA. It contains its own proteins and lipids. And you must always know that because of these two things, it is called as semi-autonomous organelle of the cell. Semi-autonomous organelle of the cell. And it also divides independently by fission. Also divides independent of the cell by fission. Which method? Fission. A method used by prokaryotes. Sometimes it also referred as endosymbiont of the cell. Okay. Now let's go to plastids. So plastids, they are only found in plants and euglenoids. So these are the organelles found only in plants and euglenoids. Plants and euglenoids. They are only found in plants and euglenoids. Very well. Now important about them is that they are easily observed as the size is large. So size is more, so they are easily observed on microscope. So these are the organelles which can be easily observed on microscope. One thing you should always remember about them. And they are also double membranous organelles. They are also double membranous organelles. And they have usually three types. There are three types of plastids. We have so-called leucoplasts. We have so-called chromoplasts. And we have so-called chloroplasts. They have different function and different role for the cell. Leucoplasts are colorless plastids which store food. Okay. Chromoplasts are colored plastids. They have varied colors which provide decoration to the plants or plant parts. And chloroplasts are green plastids because they contain chlorophyll and they help in photosynthesis. One thing you can be asked about types of leucoplasts, so that you should always keep in your mind types of leucoplasts. So leucoplasts have different types. One is amyloplasts, second is aleuroplasts, and third is oleo or elioplasts. It is depending on the type of food they store. So everyone knows amyloplasts store carbohydrates or starch. Store carbs or starch. Aleuroplasts, they store proteins. And oleo or elioplasts store lipids. This is cool. Okay. Now, most important among them is chloroplasts, which we have to discuss. So let us discuss the chloroplasts in detail. Chloroplasts. So chloroplasts, again double membranous organelles which contain chlorophyll and help in photosynthesis. So double membranous organelle which help us in photosynthesis. Okay. Now anyone can ask you about the shape of chloroplast. Okay, shape of chloroplast. So most common shape, they have varied shapes, but in mature plants they are discoid or lens shaped. So shape, they are varied shapes. They can be lens shaped, they can be oval shaped, they can be spherical in some, they can be ribbon like in some, they can be discoid, etc. But in higher plants, they are usually lens shaped or discoid. These two shapes are common in higher plants. Now, if we talk about the size of chloroplasts, so this size is, in terms of length, it is 5 to 10 micrometer. Again, it's very large size as compared to other organelles. And if we talk about width or breadth, it is about 2 to 4 micrometer. 2 to 4 micrometer. Okay. And if we talk about the number of chloroplasts, so number is also variable. Chlamydomonas, which is a unicellular alga that contains only one per cell, but her plants have 20 to 40 chloroplasts, especially in their mesophyll cells. So it is one per cell in whom, in Chlamydomonas, and it is 20 to 40 per cell in whom, in the mesophyll cells, in the mesophyll cells of plants. Okay, now again they are double membranes organelles. They have two membranes and both are smooth. So they have two membranes and both are smooth. Both are smooth, but remember, inner membrane has less permeability. Inner has less permeability. Remember this thing. Okay, now important thing, if you talk about this inner space, or what you call as stroma, this space is called as stroma. Okay, now stroma contains these flattened structures. It contains these flattened structures, contains stacks of these flattened structures, stacks of these flattened sacs, what are called as thylakoids. What are called as thylakoids. Okay, so I have a thylakoid like this, I have a thylakoid like this, and another thylakoid like this, another thylakoid like this, another thylakoid like this, and they form a stack. So here I will form another stack, I'll form another stack. Remember, this stack of thylakoids is called as granum. This is called as granum. Okay, this is another granum, this is another granum, and these grana are connected by these another type of thylakoids, what we call as intergranal thylakoids. So they are connected by these thylakoids, what are we call as intergranal thylakoids. They are connected by these thylakoids called as intergranal thylakoids. Okay, now these thylakoids are most important structures in the chloroplast, as the membrane of thylakoids contains, this is at one thylakoid, its membrane, this is the membrane of thylakoid. This membrane contains photosynthetic pigments in the form of chlorophyll and other pigments, carotenoids. So photosynthetic pigments, and what are the collections, their collections called as, their collections are called as photosystems. Their collections are called as photosystems, in which the principal molecule is chlorophyll, and this is the site where the light reaction occurs. This is the site where light reaction of photosynthesis occurs. Okay, this is most important thing that you must remember about these things. Now important thing you must remember about the number. So if we talk about number of thylakoids, number of thylakoids varies from 2 to 100 per gram, and if you talk about number of grana, it varies about 40 to 60 per chloroplast. Chloroplast. Now, so these are, for example, these grana, these are grana, these are grana, these are and these are intergranal thylakoids. Okay, now the space of stroma outside these thylakoids, what does that contain? The space around the thylakoids, space around thylakoids, that is the stroma. What does that contain? That contains same entities which were found in mitochondrial matrix. That contains first of all these enzymes, but enzymes of dark reaction. Enzymes of dark reaction. There are two reactions in the photosynthesis: light reaction, which occurs in the membrane of thylakoids, and direct reaction, which occurs in the stroma. There are 70s ribosomes. There is double-stranded circular DNA, and there are other proteins, etc. And again, this organelle becomes semi-autonomous because of these things. Because of these things, it becomes semi-autonomous. Okay, this thing you should always keep in consideration. Okay, so these are all the things, important things that you should remember about the plastids. Now let's, so chloroplast we have discussed it in this section. Now we have to discuss this structure called centriole. This structure called centriole. Okay, before centriole, I think you must discuss about ribosomes. Ribosomes may be skipped in this lecture. Ribosomes, they are also called as pallid bodies. They are also called as pallid bodies. Pallid bodies or protein factories. They are also called as pallid bodies or protein factories. Why are they called pallid bodies? Because they were discovered by George Pallid. Discovered by George Pallid. In which year? 1953. And why are they called as protein factories? Because they help in synthesis of proteins. Because they help in synthesis of proteins. Okay, that's why they are called protein factories. Now, important thing about them, they are amembranous structures, and these amembranous structures are made up of two things. They are made of only two things: rRNA and protein. That's why some people like to call them ribonucleoprotein complexes. Ribonucleoprotein complexes. Okay, and they are classified on the basis of their sedimentation coefficients. They are classified on the basis of their sedimentation coefficients, or what we call as Swedberg's number, which actually measures the density into two types: into 70s ribosomes, which are less dense, and 80s ribosomes. Okay, so 70s ribosomes, they are found in prokaryotes. They are found in prokaryotes. And 80s ribosomes, they are found in eukaryotes. They're found in eukaryotes. Okay, and they also have two subunits. They also have two subunits, and they also have two subunits, and everyone knows these two subunits. One is the 50s larger subunit, and second is the 30s smaller subunit. And here we have 60s larger subunit and 40s smaller subunits, and they help in protein synthesis. Everyone knows about that. Now let's discuss the centriole. So centriole, important thing about centriole is that centriole is only found in plant cells, and with some exceptions in the and only found in animal cell, and some exceptions are there in the plants, except bryophytes, but mainly they are found in the animal cells. Okay, so centriole is found in animal cells only. Cells only. And centriole is also a membranous organelle, which we all also discussed before in the classification. And usually there are two centrioles perpendicular to each other, like this. This is one, this is second, and they form the so-called diplosome. They form so-called diplosome, and this diplosome is surrounded by this special area of cytoplasm, which is called gynoplasm, and the whole structure is called as centrosome. So diplosome plus gynoplasm, that forms so-called centrosome. So sometimes we like to say centrosome, which is actually containing diplosome with the gynoplasm. Now what do they help in? Centrioles have two functions. One is form basal bodies of they form basal bodies of cilia and flagella, and second is they help in formation of spindle apparatus. So they form spindle apparatus during the cell division. So these are the two important functions which are performed by centriole. Now what are they made up of? They are made up of microtubules, microtubule proteins, and these microtubule proteins give a, a typical cartwheel appearance under cross section. So we'll discuss that cartwheel appearance. Okay, so this cartwheel appearance, in this cartwheel appearance we have 9+2 arrangement of microtubules. We have 9+2 arrangement of the we have 9+0 arrangement of the microtubules. Okay, we have 9+0. We have 9+0 arrangement of the microtubules. 9+0 arrangement. I will make you understand this 9+0 arrangement. So basically what we have 9 triplets of microtubules in the periphery. This is one triplet, second triplet, third triplet, fourth triplet, fifth triplet, sixth triplet, seventh triplet, eighth triplet, and I will accommodate here the ninth triplet. We have 9 triplets of microtubules in the periphery, and we have a central proteinaceous non-microtubular structure which is called as hub. So this is a peripheral triplet of microtubule, and this is a central non-microtubular hub tubular proteinaceous structure called hub. Okay, now there are these 3 triplets. In these triplets we call this A, we call this B, we call this C. So A, B, C, these are the names to individual microtubules. Now these microtubules are connected to hub by these proteinaceous structures, what we call as spokes, and is exactly these spokes which give the appearance of cartwheel. So what are these structures called? These are called spokes. And then we have these adjacent triplets which are linked by these Ca linker proteins like this. So what are these called as? They are called as Ca linkers, and this is exactly what we call the cartwheel structure. Okay, there's nothing more that you will be asked. Just remember there are 9 triplets of microtubules in the periphery, and there is a central non-microtubular hub, and hub connected to triplets by spokes, and triplets connected to each other by Ca linkers. Okay, so this is all about the centriole. Now cilia and flagella. Cilia and flagella are used for cellular motility. So they are the structures used for cellular motility. Structures, or we can also call them outgrowths of the cell, which help in cellular motility. They have same basic structure. So their basic structure is bilkul similar, very same. But there are certain differences. So we'll list these differences. There is this cilia, there is this flagella. What is difference? So cilia are usually more numerous. They are more in number simply. But flagella is usually 1, but can be 1 to 4. Another important point, cilia is a short outgrowth, while flagellum is long. And cilia beat coordinately. They coordinate with each other, while there is independent beating of the flagella. It doesn't follow a particular rhythm with respect to other flagella if present. Another important point, this cilia, this usually helps in movement of surrounding fluid or cell, while this flagella, it helps in movement of the cell itself. For example, sperms have flagella, and for example, trachea has cilia where the mucus is moved. Okay, now if we talk about this structure, be it cilia or be it flagella, they have three parts. So if we talk about cilia or flagella, they have three parts. Okay, this is one cilium or flagellum I have made. The three parts include, there is the first part which we call as basal body. Okay, there is second part which we call as basal plate, and there is third part which we call as shaft. So this part inside the cytoplasm is basal body. This part at the level of cell membrane is called basal plate, and the whole part outside is called as shaft. Basal body is formed by centriole. Basal body is formed by centriole, and it performs the function of anchorage. Formed by centriole, and it anchors, yeah, anchorage, I will say better terms, anchors. It anchors the cilium or flagellum with cytoskeleton. So it forms a base and it stabilizes it. Basal plate, and important thing, it has same structure as centriole. That is 9 plus 0 cartwheel arrangement. Basal plate is the area where the structure is changed. So it is area of transition from 9 plus 0 to 9 plus 2. And the shaft has two things. There is inner microtubular core called axoneme, and that axoneme is covered by this plasma membrane. Shaft has two things: axoneme or the core, and plasma membrane or the sheath. Okay, plasma membrane or the sheath. Okay, now this axoneme, if we check the structure of axoneme, it has 9 plus 2 microtubular cartwheel arrangement, and we are going to study that 9 plus 2 cartwheel arrangement. Okay, so in that 9 plus 2 cartwheel arrangement, which is drawn on your book also, we have these two central singlets of microtubules and 9 peripheral doublets of microtubules. So 1 2 3 4 5 6 7 8 9. So these are 9 doublets. So there are 9 doublets at periphery, and 2 singlets at center. Okay, these singlets are covered by central sheath, and they are connected by proteinaceous double bridge. So they are covered by central sheath. This is called central sheath, and this is bridge, proteinaceous bridge. Again, these, what we call as these doublets, this is a doublet. These doublets are connected to sheath by means of spokes, radial spokes. So these are spokes, and adjacent doublets are connected by these BA linkers. They're connected by BA linkers. These are BA linkers, which is quite similar to centrioles. But here is BA linker, there was C linker, because only two are there, A and B. So C is not present, so we have BA linkers. Okay, that's all about the cilia and flagella that you have to remember, and all your questions will be solved based on this. It can be a diagrammatic question or this database question. You can solve all of them. So 9 doublets at periphery, 2 singlets at center, and spokes connecting the doublets to sheath, and B linkers connected the doublets with each other. Okay, that's all about them. Okay, And at last we have this nucleus. Okay, so nucleus is the largest cell organelle. Let me tell you about this nucleus. It is the largest cell organelle. Largest cell organelle, and everyone knows it was discovered by Robert Brown. Who discovered nucleus? It was Robert Brown. And when did he discover it? In 1831. Okay, and important thing, if we directly study nucleus, usually if we talk about the number, usually there is one per cell, but some may have more than one, which is called as Centriole condition or multinucleate condition. For example, skeletal muscles. Or some may have zero nucleus, no nucleus. So these are called as denucleated cells. Denucleated cells. For example, typically you are asked RBCs and sieve tubes. RBCs and sieve tubes. Okay. Now if we talk about shape and location, which is important. So shape is usually spherical, but they can be flattened, and they are usually present at geometrical center. But some cells have peripherally shifted nuclei. For example, epithelial cell. For example, these mature plant cells, pad cells, etc. Present at geometrical center. But we'll remember what happens usually. So shape and location spherical, present at the geometrical center. Now important. Directly going to the structure of nucleus. So nucleus has this membrane which is called as nuclear membrane or karyotheca, and then it has this matrix inside, nuclear matrix, which contains several things. Nuclear matrix which contains this network inside called chromatin, a dense structure sometimes present called nucleolus, and the fluid, surrounding fluid, which is called as nucleoplasm. So I'm going to label all these components. This is nuclear membrane. This is nuclear membrane, which is also called as karyotheca. Let us remember this is a double membrane. This is a double membrane, and this double membrane has the space in between, which is called as perinuclear space, and your book has mentioned the diameter of perinuclear space, which is 10 to 15 nanometer. 10 to 15 nanometer. So this is perinuclear space. You must remember this is perinuclear space, and it also has nuclear pores, which allow transport of materials inside and outside. So I cleared all the things about this nuclear membrane. So nuclear membrane is also called a karyotheca. It's a double membrane, and there is space between the two membranes that's called a perinuclear space, 10 to 50 nanometers in diameter, and this membrane also has nuclear pores which allow the transport of materials. Then there is this nucleoplasm. Nucleoplasm is the fluid inside this nucleus. It is the fluid or matrix inside this nucleus. It is the fluid which contains uh, ribonucleotides. It contains, simply I will say, nucleotides, deoxyribonucleotides, both ribo and deoxyribo. It contains this enzymes of replication and transcription. It also contains uh, ATPs and other proteins. Okay, then we talk about this network. This network is called as chromatin. This network inside the nucleus is called as chromatin. This chromatin is stained by basic dyes. It was discovered by this Fleming. This chromatin network inside the DNA, it was discovered by Fleming. This chromatin, it was discovered by Fleming, and it is stained by basic dyes. You must always remember it is stained by basic dyes like histocarnine. Okay, basic dyes. And this chromatin is nothing, it's actually the network of DNA. network of DNA, loosely packed DNA. DNA in which phase? Interphase. It is network of interphase DNA. Interphase DNA or loosely coiled DNA. Okay, loosely coiled DNA. And this chromatin, when it is packed densely, when it is packed densely during metaphase, during metaphase, it forms so-called chromosomes. It is the thing that forms so-called chromosomes. Okay. Now we notice that chromatin has two types. It has two types. There is so-called euchromatin. You can be asked about this. And there is so-called heterochromatin. Now I cannot show you in this figure precisely because I have made it with different colors. It will be jumbled figure. I will tell you like this: heterochromatin and euchromatin. What is the euchromatin? It is a lightly stained chromatin. When you stain it with basic dyes, the area which is lightly stained, it is euchromatin, and the area which is darkly stained, it is heterochromatin. Actually what happens, this one is loosely packed as it is transcriptionally active. It is transcriptionally active. It more often performs transcription, while this is transcriptionally inactive. While it is transcriptionally inactive. Okay, so it can be asked about transcriptionally active chromatin and transcriptionally inactive chromatin. Okay, heterochromatin. X chromosome bar body is a type of heterochromatin. You must always remember. Okay. Then there is this final structure called, sorry, there is this final structure called, there is this final structure called nucleolus. Okay, nucleolus. Nucleolus was discovered by Fontana, and this nucleolus is also an amembranous structure. It doesn't have an independent membrane, amembranous structure, and it's a site. It's actually a site for synthesis of rRNA. That's why it is called a ribosomal factory. It's also called ribosomal factory. Okay, that's all you have to remember about this nucleolus. Okay, so that's all what we have to discuss about the nucleus. One final thing about chromosomes. Before I thank you, I will discuss something about chromosomes. So chromosomes, again, let me recall that it is tightly packed form of DNA, most tightly packed form of DNA, which is usually observed in the what we call as metaphase. Okay, if we talk about each chromosome, metaphasic chromosome, we talk about each metaphasic chromosome that has X-shaped structure, which has two halves. I will show you like this. This is a metaphasic chromosome, and it is having two halves. It's having two halves. And what are these two halves called as? These are called chromatids. It has these two halves. These two halves are called as chromatids. These two halves are called as chromatids, and these chromatids are united at this point. This point is called as centromere. This point is called as centromere. So these two halves are united at this point called as centromere. Okay, united at this point called as centromere. Okay, now actually these are identical DNA, these two chromatids, and they are to be separated at the anaphase. So these two chromatids are separated at the time of anaphase, you must remember. Now this centromere, it has thin protein discs on either side where spindle proteins attach. These thin protein discs on either side are called as kinetochore discs, kinetochore proteins. And what is the significance of kinetochore proteins? They help in attachment of spindle fibers. You must always remember this. Now, based on position of centromere, we have four types of chromosomes. Four types of chromosome. Okay, you must always remember this. Four types of chromosome. We have metacentric, we have submetacentric, we have telocentric, and we have acrocentric. And let me show you this by diagram. So this thing is your metacentric, this thing is your submetacentric, this thing is your telocentric, and this thing is your acrocentric. So what is the difference? The centromere is exactly at center. Centromere is exactly at center. What is about this thing? Centromere is away from center, little away from center. What is about this thing? Centromere is at the terminal, and centromere is little away from the terminal and acrocentric chromosomes. That's all that you have to remember. And the thing that has been exposed, which is the final thing. Thank you.